All right, here we are again. Oh, did I press record? Yes, I did. Good. Embarrassing. So let's talk about what the T cells do and how do they get a window into the infected cell. Now, I just want to remember that Craig was my grad school advisor, and I'm going to talk a little bit about my grad school work, which I did over at UW. He actually passed away early. If you look at the ages, he was only 60, and it was fairly recently. He was not at UW anymore. He moved on to the Medical University of South Carolina. But of course, I do want to say that this all started with him. I was not into immunology at all until I saw this project. And I wanted to do something that would use chemistry to really help people. And that's what Craig had the idea, you know. So I joined onto his lab and I copied him, you know. So I just want to say getting into a good lab, the lab that's right for you, is really important if you're making these decisions. And I'm always happy to talk about what I've learned about the right way to make those decisions, or choosing medical schools, or choosing even what kind of program you want to go to. All right. So let me ask you something I did not know, and in fact, I've only picked up this stuff on the way as I've had to from reading papers. I've never had a class in this. So does anyone know where your thymus is? Can anyone point to your thymus? Right? It's right in the middle of your throat. What happens if you remove it? Maybe you know someone. Do they ever have to do thymectomies or something like that? Have you heard of that? Yeah, thyroidectomies. Okay, thyroidectomies. But what cases have you heard about where that's needed to be done, and what's the result of the patient? Yeah, you're probably immunocompromised in some way if you don't have your whole thymus. I've even heard that the appendix, for a long time they didn't think the appendix was important, but they found out it has an immune function of some sort. Yeah. Awesome.
Well, we learned knowledge that the thymus is where the T cells, quote unquote, mature and are developed, where the receptors are developed. And so I would expect that if you remove it, the T cells that will be made maturing over time.
Right.
But we also talked about how the thymus over time gets smaller and smaller until, and so it's kind of part of how, as you get older and older, your immune system gets weaker in a sense because it's producing less stuff.
Yeah, and more imbalance. And there's a direct connection to this, to COVID, and the fact that this is connected somehow to the fact that younger people can deal with COVID a lot easier than older people. Again, we're talking population levels, not individuals. But there's the IL-6 storm is much more likely to happen with an older system. And it has to do with the immune cells not functioning as well. Yeah. It's basically boot camp, or it's school, college for T cells, where they learn what is me and what is not me. Or more precisely, they learn what is me. So when they see something else, they say, oh, that's not me. Time to attack, okay? So there it is. This is something, and here's all these biological terms that honestly, for me, I just want to look at the molecules. But there's a lot of good history in here. And by the way, if you have the chance to take immunology, take it. If you don't have the chance, reading this article is at least something. The thymus was named because it resembles a thyme plant. And you can take it out and the organism doesn't die right away, right? And in sterile laboratory conditions, you might not even notice because cellular immunity is not as important. But then there was an important study with mice where if you imagine how small your thymus is, imagine how small a mouse thymus is, okay? But this one scientist figured out how to do it. And then they noticed that the lymphocytes dropped dramatically, the T cells basically disappeared, and that's how they were called T cells, because they were connected to the thymus. So they have a receptor on their surface, just like the B cell receptors that look like antibodies, and just like antibodies. And in fact, it's even made out of IG domains, which are those beta sandwiches, and it's made of 2 chains. And it looks a lot like the antibody. It even looks like a FAB fragment, this variable fragment of the antibody. You see it's almost exactly the same size, and it has an antigen binding site down there. Now, this is what they do when you look at them under the cell. When a virus infects a cell, it, of course, is inside the cell. That's the whole point. It's taking over the ribosomes and things like that. And then it's producing more virus. A cytotoxic T cell will walk up to a virus-infected cell. It'll form this interface. You see this flat level right here. And then it will kill it. It will basically detect something's wrong inside this cell. So then it will release the hounds or whatever the thing is that it releases. I'll show you what it releases in a second. So if they can look inside to see virus proteins, they can also look inside to see tumor proteins. So there are some T cells that will recognize and react to new things, new proteins that are made inside the cell in the case of cancer. If the cancer cell is full of these new types of proteins, it gets on the surface, the T cell can recognize it, and then it can become activated. If they can kill a virus-infected cell, they can also kill a tumor-infected cell. It's full of the cancer-related proteins. And so there are such things as cancer vaccines. Cancer is a distortion of the regular cell, but it is a lot like a virus in some ways. So how do they see it? When a virus infects a cell, this is the key. When the viral proteins are synthesized in the cytosol, they go through the rest of the cell, and in the endoplasmic reticulum, there's this yellow protein waiting for them. And of course, the virus is making a ton of new viral proteins that some of them are forming virions and leaving the cell. In the meantime, some of them end up stuck to these yellow proteins. The yellow proteins get moved to the surface, and there you have the T cells, whose job is to look for the yellow proteins and to see if they're holding something new in them. If they're holding something new, then they activate. So here's a case where it's seeing something red that it hasn't seen before. And notice how the receptor works. It's binding 2 things at once. It's binding both the yellow thing and the red thing. Question?
So this MHC, is this only for immune response, like for the immune system, basically built into all cells? So that means just check what's inside?
Right. MHC Class 1 is in all cells. MHC Class 2 is in limited types of cells. But this is the first one. This is like every house has a mailbox. Every cell has this immune system in it. There are probably a few cells that don't. I'm not sure erythrocytes do. But most functional whole cells, that kind of thing. So this is the armed. It's armed. It has in Britain, it has a little baton or whatever to be able to apply force to the cell to destroy it. Hopefully it's making that decision well and justly. These are the weapons that a cytotoxic T cell has. This is David Goodsell's picture of it. And you can see there's a receptor engaged over here. I believe that this right here in red, in fact, if you look over here, you can see the little bright red peptide. This is MHC Class 1, and here you have a blue T cell receptor that's interacting with it. It's activated, and it has this whole thing of toxic materials. And you see the toxic materials, what do you see them doing? Punching holes in the membrane, letting things in. This purple thing is probably a toxin of some sort. And they can do things like make reactive oxygen species, basically make nasty things that will be unleashed against this yellow target cell. So some of the names of the stuff, there's perforin, which perforates cells. It's a protein that does that. There's granzymes, which make nasty things. And then there's reactive oxygen species that are made, because why not use oxygen's own reaction? And I just have to make that little connection because oxygen is always one of the themes that we've been talking about. And we can even get a picture of what the supramolecular attack particles are. This is what it looks like, and they they propose that there are these extracellular killing entities. So not just being like little vacuoles that you open up. Sometimes these are like long-range artillery that it sends against other cells that can actually go outside a cell and sort of like land onto the membrane and then like deliver their payload. This is a little more shaky than the other ones. The intracellular is well established. This is not as well established, but I think it's a cool idea. The idea that they can send out these little bombs that like float around until they find something in the area. But it also seems like a greater risk of inflammation, a greater sense of risk of autoimmune reaction. So that's how class 1 works, and class 1 is on most cells. There's another class 2 of MHC, and this is only in particular cells. These are in cells like macrophages. Remember that macrophages, they go out looking for trouble. And so they take in the extracellular bacterium or whatever, and they chew it up, and they acidify it. And waiting inside the acidified endosome, you have this yellow molecule. But you might notice this one has 2 tails instead of 1. Other than that, it looks exactly the same, but 2 tails means class 2. And so class 2 is going through, and it's class 2 is binding and going to the surface where it can be recognized by a different kind of T cell. And in fact, on B cells, you actually have the same kind of thing. This is before clonal expansion takes place. If a B cell receptor, which looks exactly like an antibody, binds to an antigen, it's internalized, chewed up, put on MHC class 2, because this will send the signal to the helper T cell that something new has been seen. Therefore, this cell should make more of these blue antibodies. So MHC class 2 is associated with T helper responses. Both these macrophages and the B cells, you don't want to kill them. You don't want it to be the same system as the MHC class 1 because it's their job to find these things. You don't want to reward them by unloading a bunch of perforin on them. The whole point is that you keep them alive so that they tell you what they know. And that's what T helper cells do. T helper cells sense that there's something new on the surface of the MHC class 2. And so in the case of the macrophage, they will say, hey, get activated, start inflaming, call the rest of the immune system. You've got a problem here. To the B cell, they'll say, get activated and make more antibodies, just like what you're doing. What you're doing is great. Keep doing more of it because we need more of these antibodies. Because I have never seen this little red peptide before. So they're more like the radio, the communicators that call in the other responders. They don't have cytotoxic granules. You don't want them to have it. Both of these have similar T cell receptors. All the T cells have a T cell receptor. It looks like this. But I want you to compare it to what you know about antibodies. Well, first of all, before you compare it, let's just look at this. Look at its domain structure and tell me something that you observe about its domain structure. What can you tell by looking at this? What is true about a T cell receptor structure? Well, you can't, there's a disulfide bond here, and I think that the antibody has a different pattern of disulfide bonds, so they're close enough to make a disulfide bond. I'm not sure you can draw any conclusions from how separated they are, but I mean, the thing is they have an interface. That's basically the level at which we can say things. What do you see?
The variable region is at the top, and that's where the high is.
Right. So you have like this surface, this weirdly shaped surface. You have 2 variable domains on the top. And what else do you see? It's actually, what we're going at with this one is pretty simple. There's a little carbohydrate. It's on the outside rather than the inside. Like most things, it's glycosylated. And just go to the bottom, and you got 2 transmembrane regions. You got 2 tails for it. This is the T cell receptor. So it's membrane bound. It's heterodimeric. It's got an alpha chain and a beta chain. They're the same size, but they're different chains. They're different colors. And it has variable and constant domains, just like antibodies. It's just it has 4 rather than 12. And in fact, if you look at it, so if you look at it from this perspective, The secondary structure looks just like an antibody fragment. It looks just like 4 antibody domains stuck together. You look at it at the end, and you also see hypervariable loops, just like with an antibody. In fact, there's even 6 of them with 2 extra ones on the side that we debate whether they're important or not. There's 6 main loops, just like with an antibody. And this is what they've known. I'm actually showing you some of the first crystal structures, because when I was in grad school, when I started grad school, we didn't have any crystal structures of these things. And they came out as I was in grad school. So it was really great to be able to write a dissertation with these. So that's the T-cell end of things. On the target cell end of things, you have the MHC. Here's an example of an MHC molecule. And it looks kind of like an antibody surface that's bound to peptide. You have, this is the MHC protein. And by the way, the genes are called the HLA genes. And so you can see either term. It's one of the things, remember, immunologists never want to use the same term twice. But so MHC and HLA are essentially equivalent for our level. But it's holding this peptide that is from the inside of the cell. By the way, I can tell this is class 1, but that's because I know what to look for right now. So it's like a window into the cell. This red thing was made inside the cell. Or this, it reminds me of a hot dog in a hot dog bun. This is like one of those little 7-Eleven hot dogs, the cheap ones. But if you spend more, you get the hot dog that is bigger than the bun. And that would be actually the class 2 MHC. So the big bite is the MHC is the class 1. And the super foot long is the MHC class 2.
How were the proteins selected to be taken up out of the cell for the MHC type 1?
Well, you have 6 of them in your genome. You have HLA A, B, and C, and you have a copy of the gene. You get one from each parent. So once in a while they match, but usually they're different. So usually you have 6 different copies of this. They're all hanging out inside the cell, and you have these when viral proteins are overproduced, you have just some proteases inside the cell that just chop everything up, and you end up with a huge complex array of things that end up binding and going to the surface. Mm-hmm. It's whatever in your genes. And so you will have 6 spaces. You have 6 versions of this that are slightly chemically different. They have different grooves, and they have different surfaces. But your T cells have learned to recognize all those surfaces. So your T cells are used to your 6 proteins and not to other people's. So here's MHC class 1 and class 2. This is from the side. And you can see the peptide goes up there, and it sort of fits. This was the top view, and this is a side view, okay? So you see that underneath the top view, you have yet more IgG domains, except this time you only have 2 of them rather than 4 or 12. But at least, there are some things in structural immunology that are easy. Everything uses Ig domains over and over again. So I want you to compare the domain structures. Here's the one of TCR. Here's the one of class 1, and here's the one of class 2. And I want you to, compare them. This was heterodimeric, but heterodimeric of 2 equal molecular weight chains for TCR. In those terms, what do you see is the same or different from the other ones? And 2 transmembrane regions. So MHC class 1 or MHC class 2, how are they the same? How are they different from TCR? Yeah, and actually, this is probably, it's less specific in general, and that's something that you would have to look in more detail to be sure about. But this is actually accurate. That's an accurate conclusion from it. MHC class 2 actually have a bit wider of a specificity than MHC class 1. It's a bit narrower and a bit choosier. MHC class 1 is a little bit choosier. And that's sort of the advanced thing that you can take away from this. Even going more simple than that advanced conclusion, what can you conclude about how many chains there are and how many domains there are in each chain?
There's 3, there's 8, 1 through 3.
Mm-hmm. Right. I mean, this is heterodimeric truly, just class 2 is heterodimeric like the TCR is. Class 1, There's only one long chain with 3 domains, and then you've got this extra domain which doesn't even go through the membrane, beta-2 microglobulin, which is just a little Ig domain. It's actually pretty cool because you can isolate it and you can float it around. You can use it as a reagent because it's pretty darn stable. But yeah, it's, they have fundamentally different membrane structures there. Yeah. I think that's, oh, the other thing that you can see if you look at the top, the alpha-1 and alpha-2, the alpha-1, beta-1 domains, those are not Ig domains anymore because they have peptide binding clefts. And so they are very different. In fact, you can see up here, Ig, and up here you have alpha helices for the first time. Because the alpha helices are the 2 buns for the hot dog, okay? I think I actually said this in my graduate dissertation defense, and so I have to always, it's like the one thing that everyone can understand. Everyone knows what a hot dog looks like, okay? So you have it looking from the top. You see it like this. This is class 1. And it binds peptides like this. The one thing you'll notice about it, the ends are closed in. And so if you take 3 different peptides that bind the same class 1 protein, they're each 9 to 10 amino acids long. Half of them point up, half of them point down. Some of them point to the side, but let's not get too detailed. You notice that the pointing down ones are all the same. The pointing up ones can be very different. But all of these have a leucine here pointing down and a valine there pointing down. There's a pocket in the MHC, and you can infer that that pocket is hydrophobic. It can accommodate, it's the right size and chemistry to accommodate leucine or valine. It won't bind peptides that have like an arginine there. It just can't fit. But all these surfaces can be very different, the surfaces pointing up. So in fact, you can look at what peptides bind different MHC class 1s, and you can see they have different specificities. They care about different things. But they do have, there's always a pattern. You can see that this one likes to bind tyrosines at position 2. This one actually has a pocket that cares about something in the middle, and it cares about this, this leucine all the way to the C terminus. So you look for these things, and you look for the things that are common. The common parts are going to stick down into the protein. The other parts are very variable, and they can present a variable surface. So how does it stick in? And this was the topic of my graduate research. I wanted to look at how important were the different hydrogen bonds. Now this is MHC class 1, which I did not work on. MHC class 1 binds the ends of the peptide because it has ends, and remember it's closed at the ends. So it binds the peptide at each end, and it also has pockets that accommodate the things going down. MHC class 2, on the other hand, from this perspective, it looks pretty much the same. But if you look carefully at it, the peptide binding cleft is a little bit wider, just like Leanne noticed. And it's actually open at both ends. So this one can have the hot dog that goes out the end of the bun. In fact, you can see this. You can compare class 1 to class 2. The class 2 can even have different lengths of ends dangling off the side. And so, if you look at class 2 proteins, they have all different lengths, and it's actually a little bit harder to find the anchor residues. But here we've lined them up so that you can see you have a negatively charged anchor at position 4 and a hydrophobic anchor at position 9. This is actually the mouse molecule. I actually worked with this molecule. Maybe one of these peptides, once I would used to be able to know the sequences, so I'd be able to tell you exactly which peptide I worked with. I'm pretty sure I worked with one of these. So MHC class molecules, they form hydrogen bonds all the way up and down. They can't hydrogen bond the very ends. So they form it from side to side. And my question as a graduate student was, we had the crystal structure of this, and all these hydrogen bonds look about the same. But are they all the same? If we mutate them, does one type of hydrogen bond matter more than the others to the stability of the complex? And it turns out that these two, I still remember this one. See this one and this one? There's even symmetric ones at either end of the groove. They look a lot the same to you, right? Actually, if you mutate this one, it really destroys stability. If you mutate this one, it just kind of shrugs and moves on. So that's what we found out. We used in chemistry in our lab, and we said this end of the molecule was more important. And my big accomplishment in graduate school is that later, a few years later, another laboratory came along, found the exact way that this whole system works, and it turns out there's a protein that sort of shuffles the deck. It opens up the MHC, and it lets peptides move in and out, so you have them more shuffled. Deliberately destabilizes it. That protein binds right here. So I gave the biophysical reason before they knew for sure that they did it, and then they cited me in the paper. So that is the one thing that I can say that my dissertation actually had an impact on other research. So this is my dissertation, 2001, long time ago now. And I compared the N-terminus to C-terminus hydrogen bonds. It turns out that the N-terminus is much more important. Sandra? Yeah.
Is it that type of variable feature that is going to be different between both T-cell receptors, or between like individuals that have different MHC?
Well, these sites are conserved, actually. You want the hydrogen bonds, the hydrogen bonding residues are going to be conserved. What's not conserved is the surface of the helix that's presented to the T-cell receptor, and especially what's not conserved is the pockets underneath. So it's like underneath is not conserved, above is not conserved, but this level right here where you have all the hydrogen bonding, that's much more conserved. And so you can actually, you have some differences, and you can have differences from peptide to peptide, but in general, you're making a bunch of hydrogen bonds equally across it. Yeah.
And out of curiosity,
Yeah.
how did you measure the usability of the peptide MHC complex?
We didn't go for equilibrium, but we actually just measured off rate. So we were as chemists because our reasoning was that on rate doesn't matter anyways because it's not physiologically important. It's also a pain in the rear to measure. So we just measured off rate. And I can tell you a lot more about how I did it, but we measured kinetics. And in fact, if you look at my title, I do say cooperative energetics of binding interactions, but I should have said kinetics. I know that in my abstract I said kinetics at one point, because technically we were not looking at thermodynamics. But life is not at thermodynamic minimum anyways, so kinetics is sometimes what's important. Yeah, so this is the crystal structure of the TCR-MHC interaction. And you can see that the 6 loops land right on top of the peptide. They recognize both the alpha helices of the MHC and the peptide surface, but only the surface is pointing up, of course. Surface that's pointing down doesn't matter as much, to the T cell at least. And I know this is really fuzzy, but this is how I first saw it. This is they actually got a crystal structure of the TCR-MHC with peptide in the middle for class 2. And so I was very happy to see that. Now, of course, we've gotten a ton more structures, and there's a ton more kinds of immune system things. And you can see that they all have their own little footprint that they land on the MHC. And some of them bind more peptides, some of them bind less peptides, some of them bind at this end, some of them bind at that end. There's a whole bunch of stuff going on. But the one thing I want to make sure you don't miss, redundancy of ligand recognition. There are some that bind conserved regions of the protein, so they will bind all proteins. There are some that don't want to bind redundantly. It all depends on what kind of immune response you want. So what other words could we use for redundancy? Any ideas of redundancy of recognition? This is where the word cross-recognition comes in. And that's what, this is part of the reason why I'm so interested, because you do have these issues. Sometimes the receptor wants to bind things that are variable. Sometimes it wants to bind things that are conserved. Sometimes it wants to bind 1 MHC. Sometimes it wants to bind 10. And it's all of that is a balance, and it's really complicated. It has to do with immune systems. So this is the question I want to end on, and then we'll wrap up the rest of it online. But why would Wells run phage display on antibodies rather than T-cell receptors? He wanted to look inside the cell, right? So why not you make a single-chain T-cell receptor, like a single-chain antibody? Any ideas on why he did not do that? He had good reasons. Let me just say, you can't just say that he was silly, that he didn't know what he was doing. Why would you not do what Wells did with T-cell receptors?
Kind of thinking about cancers' ability to
Mm-hmm.
And because of that, a T-cell receptor wouldn't be able to recognize the difference.
Right.
Antibodies would be able to recognize more than just peptides that are presented by the cell. They would be able to recognize the surface and different components on that.
Yes.
That could be important.
That's a very good reason. That's one thing that you don't have to go through the antigen presentation mechanism. And cancer cell, not to mention, if a cancer cell has a mutation in a couple of places, if it's 100 residues long and it has 2 mutations in it, what are the chances that those residues are going to be in the 10 residue snippet that shows up on the MHC Class 1? Not good odds. So there's that side of it as well. Any other hypotheses? That's one of the reasons.
For cancerous agents, would they sometimes just disable their MHC generating genes?
Yeah.
Like, I won't give you these.
So a tumor cell might survive if it disables its antigen presentation machinery entirely. Now, the good news is we have mechanisms to look for that and say, hey, you don't have a driver's license, get out of here. But those are different mechanisms, and they don't always work. So yes, there's the whole thing of it can circumvent it entirely. And the other thing is, remember what I said about how many MHCs each of you has? Each of you has 3 to 6 different MHCs. That means that the antibody that would be binding is going to recognize an MHC surface as well as the peptide surface. But even if we had a single peptide that we wanted to go after, we'd have to worry about the 6 different MHC surfaces that you were presenting. And that would make it harder for our antibody to work, if it was binding any part of the MHC. Now, the thing you can say to yourself, what if I can make an antibody or a T-cell receptor binding molecule that just binds the peptide? I would have thought that you can't do that. But there's actually a group of people that did that. And they came up with a cross-reactive molecule that actually is an antibody fragment that will bind one cancer-associated peptide that's presented by multiple MHCs in multiple people. I say people, but they tested it in mice, so multiple mice at least. So we're going to talk about what that looks like and how that therapy works. That will be our example number 1 of cross-reactivity. And then I will have one more talk after that with some faster examples, 4 more examples of cross-reactivity. Probably going to take me about half an hour to get through all that, just kind of like we had. Just like an MHC Class 2 molecule, this lecture sort of dangles out the end. Okay, so we're going to have a little bit extra. But it's important, and at the end of it, make sure you do watch that pretty soon. Number 1, it will be on the quiz. Number 2, at the end,